हेलो एवरीवन। आई होप मेरी आवाज़ सुनाई दे रही है और स्क्रीन दिखाई दे रही है। मेरे एंड पर तो भाई दिखाई दे रही है। पड़ोस वाली आंटी की आवाज़ को अवॉइड करें। इतना तेज मुझे नहीं पता क्यों बोलती है कि हमारे घर तक आवाज़ आए। शी इज सो आउट लाउड। जिसे एक्सट्रोवर्ट बनना है उनसे जाके संपर्क करें, वो आपको बनाकर ही दम लेंगे एक्सट्रोवर्ट। खैर, उन्हें छोड़ते हैं और अपने क्वेश्चंस पर हम आगे बढ़ते हैं। ठीक है, विच ऑफ द फॉलोइंग इज नॉट कंसिस्टेंट विद द base pairing in a double-stranded DNA molecule? The molecule of C equals the number of G. Okay, the number of A equals the number of T. The amount of purine is equal to the amount of pyrimidines. The ratio of G plus C to A plus T is same for each species. फोर्थ वन हमें तो सही नहीं लग रहा। फोर्थ वन इज द राइट आंसर। आगे बढ़ते हैं। इन यूकैरियोट्स विच ऑफ द फॉलोइंग इज रिमूव्ड फ्रॉम इनिशियली ट्रांसक्राइब आरएनए before is it is transported to the cytoplasm for translation? Intron 3 poly A tail ribosome binding site 5 gap. Intron. Question number third. The process of splicing in eukaryotes is reminiscent of antiquity, represent dominance of RNA world, is an indicator of the complexity of human genome with the legacy of organic evolution. Is reminisce represent dominance of RNA world. In prokaryote, the predominant site for control of gene expression is the transcription initiation processing level, transport of mRNA transition level. Transcription initiation. Yes. Next question is, in Hershey and Chase experiment, what finding led to the conclusion that DNA is the genetic material? Presence of radioactivity in the bacteria with 35S was used. Presence of radioactive in the supernatant when 35S was used. Presence of भाग ही गया. Presence of radioactivity in the bacteria with 32P was used. The presence of radioactive in the super. Third is the right answer. Yes. कभी कभी इतना कॉन्फिडेंटली बोलने में डर भी लगता है कि भाई गलत हो गया तो मोए मोए हो जाएगा। अपने आ गई कि भाई खुद ही तो बोला था कि इतना सही आंसर होगा, खुद ही इतना कंफ्यूज हो कि गलत भी करके बैठे हो। Which of the following important biochemical reaction in living system is catalyzed by a ribozyme? Repair of DNA, electron transport chain, formation of peptide bond, organization of mTOC during the cell division. Ribozyme, formation of peptide bond. Yes. VNTR are useful in DNA profiling because they are hypervariable, are inherited, synthesize constitutive enzyme. VNTR are hypervariable, are in VNTR used in DNA profiling. Okay, are inherited, synthesized constitutive enzyme. I'm not sure about this, but let's go with first one, and it's the wrong one. Synthesize constitutive enzyme वो नहीं करता है, बाकी सब कुछ कर देता है. Which enzyme polymerize RNA with defined sequences in a template independent manner? Peptide transferase, RNA polymerase, RNA transcriptase, polynucleate phosphorylase, RNA reverse transcript नहीं phosphorylase. The process of RNA splicing shows the dominance of RNA world. अभी ऊपर आया था क्वेश्चन. In RNA splicing, exon are removed नहीं, intron are removed, exon are joined together. The core RNA polymerase is capable of catalyzing which step of transcription? RNA polymerase, all of these नहीं, elongation only. RNA polymerase सिर्फ elongation करवा सकता है. In most prokaryote, the transcription unit is monocystonic. पॉलीसिस्टोनिक यूकैरियोट में होती है मोनोसिस्टोनिक, प्रोकैरियोट में होती है पॉलीसिस्टोनिक. The DNA dependent DNA polymerase catalyze both the direction only in 5' to 3' direction. Reverse transcriptase is a DNA dependent RNA polymerase. RNA dependent DNA polymerase बहुत confusion होते हुए. A molecule that can act as a genetic material must fulfill all the following criteria. Except it should be able to generate its replica, it should chemically and structurally be stable, it should provide scope for rapid mutation, it should be able to express itself in the form of Mendelian characters. Third one is the not right answer. Yes, वो फॉलो नहीं करता मतलब उसको. The unequivocal proof that DNA is a genetic material was provided by Hershey and Chase. Which of the following is not a feature of the double helix model of DNA? The two chain have antipolar polarity, appearing always come opposite to a pyrimidine. The pitch of DNA is 3.4 nanometer. The two chain are coiled in a left-handed fashion. According to Irwin Chargra, for a double-stranded DNA, the ratio between AT and GC are constant and equal to 1. The ratio between AT and GC are constant but not equals to 1. The ratio between AT and GC are constant and equals 1. AG and TC. The ratio between AG and TC are constant but is not equal to 1. मेरे हिसाब से फर्स्ट. यस, सबने ज्वाइन किया हुआ है और चैट में कोई आंसर नहीं कर रहा है. आलसखोर लोग. To form a nucleoside, a nitrogenous base is linked to pentose sugar. To form a nucleoside is linked to pentose sugar through a N-glycosidic linkage at carbon atom number 1. Yes, this is the right answer. Identify incorrectly matched pair. ये तो ठीक है, ये भी ठीक है. इससे रेशियो कोलाई 4.6 होता है. Third is the wrong answer. The amino acid acceptor arm of the tRNA is as at its T5C loop, DHU loop, 5' and 3' and this question is not from NCERT at all. But guess मार लेते हैं, अब सामने आ गया है तो DHU loop नहीं, 3' end. Which of the following codon has dual role in the genetic code? AUG. What is the major difference between the lagging and leading strand during RNA replication? आरएनए रेप्लिकेशन में डिफरेंस बताना है लीडिंग और लैगिंग स्ट्रैंड. On the leading strand, DNA synthesis occur from 5' to 3', while DNA synthesis occur from 3' to 5' on the lagging strand. DNA polymerase is able to continuously add new nucleotide on the leading strand, while it must keep starting over on the lagging strand. The lagging strand replication. The lagging strand require only a single primer, while the leading strand require many helices. Open the leading strand at a faster rate than फर्स्ट होना चाहिए मेरे हिसाब से. इतनी लंबी कहानी पढ़वा, बट फायदा कुछ नहीं. सेकंड है डीएनए पॉलीमरेज एबल टू कंटिन्यूअसली ऐड new nucleotide on the leading strand, while it must be keep starting over on the lagging strand. Okay. The inducer of lack of peron is lactose. Yes. Regulatory domain of the most activators interact with the transcription factor complex, RNA polymerase repressors, DNA binding domain, the transcription factor complex. DNA chemically is less reactive and structurally more stable than RNA. Therefore, DNA has evolved from RNA. RNA can directly code for RNA is better genetic material than DNA is better genetic material than RNA. Proteins in this RNA. Third one is the right answer for this question. A transcription unit does not contain a promoter, the structure, gene, terminator, and operator. Operator. Regulation of lack of promoter by repressor is referred to as negative regulation. Negative ही है हमारे syllabus में. A synthetic mRNA of repeating sequence 5' is used for a cell protein synthesis system like the one used by Nirenberg. If we assume that protein synthesis can begin without the need by any initiator codon, what product or products would you expect to occur after protein synthesis? One protein consists of a single amino acid, three protein each consist of different single amino acid, two protein each consist with a alternating sequence of two different amino acids, one protein with an alternating sequence of two different amino acids. One protein consists of single amino acid. Three protein each consist of different single amino acid. एक दो तीन. I'm not sure about the answer of this question. Fourth, one protein with an alternating sequence of two different amino acids. Frederick accidentally discovered transformation when attempted to develop a vaccine for pneumonia. He injected mice with sample from S strain virulent and or R strain non-virulent pneumococci bacteria Staphylococcus pneumoniae. Which of the following result is not consistent with Griffith experiment? Injected S strain mouse die, injected R strain mouse live, injected heat kill S strain mouse live, injected mixture of heat kill S strain and live R strain mouse live. Last one. आगे बढ़ते हैं क्वेश्चन नंबर 31. Which scientist first gave experimental evidence that DNA is the genetic material? फर्स्ट वन ही है जाना चाहिए. बीडर एंड टेटम का कोई रोल नहीं है. फर्स्ट पे जाते हैं। एंड इट्स द राइट आंसर।